Good morning, everyone. Today we are going to discuss on tissue processing. Before starting the tissue processing, we have to know what is histology and what is histopathology. Histology is the science of examination of normal tissue. Histopathology is the science of tissue for presence and absence of change in the structure due to the disease process. The specimens received in the lab is 10% formalin, so we have to keep crossing appearance, measurement, notification, pathological, and changes, etc. are keep for the formality for formalin for fixatives, and bits give for the respective area, not greater than 4 millimeter thickness. Tissue process and final staining the star and observe in the examinations. Introduction. The process of the tissue involvement retarding the tissue film so that the section can be cut and studied under the microscope. The process are involved in the processing of the tissue are collection, fixation, dehydration, clearing, and embedding. These are the process steps of the tissue processing. We have to follow it. The definitions of tissue processing is the tissue must be undergo a preparatory treatment before going a section in trialing to impregnated of the specimen with an embedding medium to provide supporting and support suitable constituency for microscopic tissue processing. How will prepare the treatments? 10% we have to fix and alcohol. In alcohol we have to dehydrate, clearing we have to clear in the xylene, and we have to do the paraffin in the evening. Okay, these are the dehydration, clearing, and evening. These are the step of the tissue processing. In dehydration, water molecule is removed from the tissue. In clearing, we have to dehydrate agent and replaced by the clearing agents. In impregnation, the tissue implanted with a supporting medium. This is the routine method of the tissue processing we have to follow, and the stepwise is dehydration, clearing, embedding. These three are the main step for the tissue processing. In the tissue processing, we can see that we need the grated alcohols and absolute alcohols. We have to keep the specimen in the alcohol for 2 hours in the room temperatures and in the 45 degrees Celsius. And in clearing, we have we have used the reagents like xylenes for we keep for the 2 hours. And embedding we have to yeah, place in that in the paraffin and paraffin 1 and paraffin 2. Okay, for dehydration is the removals or water removes from the tissue and cell. This place is occupied by mix. Tissue sends through the grade alcohol 70%, 80%, 90%, and absolute alcohol. Ethyl methylene isopropyl alcohol and acetone can be used. Yeah, this is the process in which the water contained in the tissue can be processed is completely reduced by passing the tissue through increasing and concentration of dehydrating agents. The various dehydrating agents are ethyl alcohol, acetols, isopropyl alcohol, dye agents. These are the agents which are used for the dehydrations. How will keep the dehydration in that in the keep in the process? We have to make the great alcohol 70%, 70-95% absolute alcohol we keep for the 1 hour, 1 hour, 1 hours. And this dehydration is done so that the wax, that is the paraffin wax which are used for impregnate, can easily miscible as it is immiscible in water. In clearing, you can see alcohol from the tissues and the cell is removed, decolorization and replacing by the fluid in which the wax is soluble makes tissues transparent. Xylene, tolerance, benzene, chloroform, cedarwood oil can be used for the definitions. The steps of the flowing dehydration is called the clearing and constitute replacing and dehydrate with a substance that will be miscible with an embedding medium. The term of clearing comes from that the clearing agents for having a same refractive index as protein. What is impregnation? Impregnation is the empty space in the tissue and cells after removing and clearing agents are taken by the mountain wax. It may be harder the tissues. It is helping for cutting the section. Melting point is 54 to 62 degrees Celsius. The definition is in this tissue is keep the wax but containing multifact from 6 to 8 hours. The max is inflated and inflated of the tissue which are increase the optical differentiate and harden the tissue help easily section of the tissues. The various wax which are used are paraffin wax. These are the agents we are used in the embedding paraffin wax, paraflux, paraffin blocks, gelatin, and celluloidine. Jars are containing of molten paraffin wax. These are the containing of paraffin wax. In embedding, it is done by the transferring the medium which are have been cleared of the alcohol to mount a filter and mount with the wax of alcohol to fill the solidify. After solidify, a wax or the block is obtained, which is then sectioned to obtain rebounds. There are type of mount L-MOX. We'll see that the L-MOX shape it is called as the leukoid mode. L-shapes barrels are spacing which are the placed in opposite position can be manipulated to increasing or decreasing the side and block of the prevision glass or metal prepared discs. Was water and paper board. Thank you.